Welcome to session 2, which is all about plant physiology, biochemistry, and biotechnology. I'm not going to waste any time. Our first speaker is ready. She is A. Alimi, and she is going to talk about fungal communities associated with legumes. Lukemi, over to you. Is the presenter ready?
Hey Alimi, we've sent you a notification in Remo to accept to come up to the stage. Please just click yes and accept. Or if you did press accept, please just activate your camera and microphone at the bottom of your screen, because we cannot see you right now. So make sure your presentation is now closed. There we go. Perfect, we can see you, and now you can share your screen.
Can you see my screen?
Not yet.
Yes, we can see you. Oh, yeah, we can see your face. We cannot see your presentation yet.
Okay. Windows D. Nice. Share. Is it fine?
Perfect.
There we go. We can see your presentation.
Okay, thank you. So the title of my presentation is the first report of avascular mycorrhizal fungi communities associated with the roots of indigenous legumes of South Africa. This is the content of my presentation. Like animals and humans, plants require adequate nutrition for growth, survival, and reproduction. But then you realize that in most ecosystems, plants are usually faced with various challenges, ranging from suboptimal availability of soil mineral nutrients to adverse conditions that are caused by biotic and abiotic factors. And in order to circumvent these problems, plants have evolved morphological, biochemical, and molecular mechanisms in order to cope with these stresses. While these mechanisms have been greatly explored, the significant roles that are played by beneficial soil microorganisms have received relatively less attention. A very good example of such a beneficial microbe is the avascular mycorrhizal fungi, which are known to enhance the nutrient uptake of plants in soils that are deficient in nutrients, and may also help plants in adapting to biotic and abiotic stresses. And as a mutually symbiotic association, the plant also deposits a substantial amount of carbon into their rhizosphere, thereby enhancing the nutrition, growth, and reproduction of the fungi. So what actually triggered our interest in this study? South Africa has a semi-arid climate. Regions like this are usually characterized by low soil nutrient concentrations, acidity, and drought. That notwithstanding, the country supports a highly diverse indigenous legumes, which are widely distributed in different biomes, with 1,662 species of legumes indigenous to South Africa. The question therefore was, could the AM symbiosis be a potential adaptation mechanism of these legumes to the semi-arid condition of South Africa? Therefore, the aim of this study is to describe the diversity of active AM fungi communities that are associated with the roots of 11 indigenous legume species in Gauteng and Mpumalanga provinces, using high throughput sequencing of the partial small subunit ribosoma nucleic acid gene. The objective is to assess the AM status, the levels of root colonization, and the diversity of root colonizing AM fungi communities. And the second objective is to examine the effect of soil properties on the AM fungi communities. In order to achieve this, We sampled legumes from Gauteng and Mpumalanga provinces based on records of previous collection from these two places. We also sampled soil beneath each plant at 0 to 20 centimeter depth using a sterile auger. So to substantiate our claims on the nutrient status of the soil, we analyzed some physical and chemical properties of the soil using standard protocols. And to assess root colonization by AMF fungi, we used staining and microscopic assessment. The grid line intersect method was used to estimate the extent of root colonization. And for molecular identification of the AM fungi in the root, we used Illumina MySeq sequencing of the partial SSU RNA gene coupled with bioinformatic analysis. After that, we use appropriate statistics to analyze our data. So this is the result for the soil analysis. As we can see, the soil is quite low in nutrients, particularly in phosphorus, nitrogen. And then another thing we can see here is that the soil is acidic. So microscopic assessment of the roots showed that all the legumes that were examined were colonized by AM fungi. Typical AM structures were found in the root cortex. These structures include the hyphal coils and the arbuscles, which are known to be sites of nutrient exchange between the two symbiotic partners. Also, we found the extraradical hyphae, which would normally forage the soil for nutrients, and then the intraradical hyphae, which delivers the nutrient to the plant root. We also saw vesicles, which are known to be storage structures for the fungi, as well as spores, which are known as propagules, and they may also function as storage structures for the fungi. So for the extent of root colonization, we found out that all the legumes that were examined were colonized by AMF, and then the level of root colonization was quite high, particularly in trifolium species, whose entire root length was almost colonized by the fungi. So for the molecular diversity of root colonizing fungi communities, after quality filtering of the raw sequences that were generated from the Illumina MySeq, the resulting sequences were assigned into 172 operational taxonomic units based on 97% sequence identity. These 172 OTUs could be assigned into 8 genera, and the ones that could not be identified to the generic level were classified as unclassified at the various taxonomic levels. It is worthy of note that out of the 172 OTUs that were detected, the genus Glomus contributed to most of the OTUs. For the relationship between soil properties and the fungi communities, we observed that soil properties actually influenced the composition of the AMF communities, particularly manganese, nitrate, pH, and organic carbon were the significant soil drivers of the AMF community composition in the roots of the legumes. So we observed that the colonization of roots by AM fungi suggests that the AM symbiosis is indeed essential for the adaptation of the legumes to the semi-arid region of South Africa. The high level of root colonization that we observed may be due to the low concentration of the nutrients in the soil of the examined plant. Generally in nutrient deficient environment, plants would apportion more carbon. They tend to apportion more carbon to the fungi in order to stimulate mycorrhization for the purpose of nutrient acquisition. We also found differential colonization levels in the plant, which may be caused by the variation in the quantity and the quality of root exudate that were secreted by the different legume species. Differences in colonization abilities of different AM fungi may also be responsible for this, as well as the environmental influences. We found that the genus Glomus predominated the AMF communities, and this could be attributed to their high reproductive capacity and higher ecological adaptability. We observed that some AMF fungi could not be identified at higher and lower taxonomic rank, and this may be due to the use of the SSU gene, which is a short DNA gene that has the problem of not being able to resolve closely related species, and may also not satisfactorily define taxa at a higher level. So going forward, we suggest that the entire rDNA region should be sequenced with the third generation sequencing technologies such as the PacBio SMRT sequencing platform, as this would enable a higher taxonomic resolution. We also found that manganese, nitrate, pH, and organic carbon were significant drivers of the communities. Studies have actually shown that the soil environmental factors are potential drivers of AMF communities. In this case, soil nitrogen may suppress or enhance the AM fungi sporulation and abundance, and this would ultimately affect the communities. Furthermore, soil organic carbon serves as energy source for proliferation and spore production, which would also ultimately affect the communities that were detected. The presence of AM fungi can improve the uptake of manganese, which are critical for nitrogen fixation. So all these could justify why manganese, nitrate, pH, and organic carbon were significant drivers of the community. So to conclude, we have seen that this study has shown the ecological significance of the AM symbiosis on the legumes, as seen in the high colonization ability and the diverse AM fungi communities that were sustained in the roots of this plant. This study has also contributed significantly to the biodiversity of AM fungi that are indigenous to South Africa. We found that glomos predominated the communities, and as such, this genus could be the most promising genus for use as inoculant when investigating the ecological impact of the interactions between AM fungi and the studied legumes. Given the numerous positive roles that AM fungi could have on plant growth, fitness and productivity, we recommend that future studies should conduct greenhouse studies to investigate the symbiotic effectiveness and the responses of the legumes to inoculation by different AM fungi, isolate or combinations. We also recommend that more indigenous legumes from other biomes should be assessed for their AM fungi diversity, as this would deepen our understanding of the legume AMF symbiosis and facilitate the exploitation of the symbiosis for the sustainable propagation of these legumes. I would like to acknowledge the following people, and many thanks for listening.
Okay, thank you, Afulukime. Are there any questions from the audience? I don't see any questions in the Q&A box. Maybe I can just start off by asking, in the trials that you have done, how much carbon was in your soil? Can you hear me? Yeah. In the trials that you have done, were there a lot of carbon in the soil, or were there little carbon in the soil?
You're asking about carbon?
The carbon in the soil, yes.
Hello?
Yeah. Yes. Were there a lot of organic material in the soil, or were there little organic material in the soil?
Yes. At least to a certain extent, the organic carbon was higher compared to phosphorus and nitrogen.
Okay.
So out of all the, the primary nutrients, phosphorus and nitrogen were quite low in the soil. So,
so
but the organic carbon was a bit higher.
Okay. If you increase the organic content of the soil, do you think you will get a better reaction from the fungi or not?
Well, for the fungi to also function optimally, they have there's a specific level of soil nutrient concentration, because if the soil nutrient concentration is too high, then it can also suppress the establishment of the symbiosis. So there is an optimum level of carbon that has to be in the soil at which it can either suppress or enhance mycorrhization.
Okay. Okay, thank you very much, Afulukime. I don't see any other questions. Okay, yeah. So you can stop sharing your screen, then we can continue to our next speaker.
Okay, thank you.
All right. Our next speaker is Danielle Botha, and her talk is all about database for food identification. Okay, there's Danielle. Right, Danielle, I can see you.
Okay. Okay, I will try to share my screen now. Is it in full presentation mode now?
Okay, just a second. Yes, I can see your presentation.
Okay.
You may proceed.
Okay. Hi, my name is Danielle Botha, and the topic of my presentation is an RBCL and tRNL reference database for the identification of food items from herbivorous diets based in the South African savanna. The research was completed with the help of my supervisors, Professor Chandra Barnard, Professor Francis Siebert, as well as Dr. Mornay du Plessis. I'm sorry. The research I'm presenting is an important aim I have fulfilled for my dissertation, which is about bioinformatic filtering options in herbivore dietary analysis following a DNA metabarcoding approach. In order to identify traces of DNA left in the feces of herbivores, one firstly needs an accurate DNA reference database to compare the unknown sequences against. This DNA database is compiled by acquiring species lists representative of a South African savanna, which will be used to download their corresponding sequences. I wanted to start with this figure by Siebert and Dreber to illustrate the incredible diversity we can find in South African savannas. Imagine being able to take a sample of soil or herbivorous feces and characterizing specific components of the plant community. This is the idea behind environmental DNA, or eDNA, and that all living organisms leave traces behind in their environment. But how do we identify an organism from an environmental sample? This is where DNA
But it seems like we have some problems.
Yes, I think it's connectivity, sorry.
I was wondering, is it her connectivity or is it my connectivity? I wasn't sure.
No, it's definitely her connectivity. Would you like to proceed to the next presenter that we have online, which is
Makatini.
Makatini's
Sorry?
not here, but the next person that we have online is Mendi Letsoalo.
Okay, I think let's continue with Mindy then.
Thank you.
All right. Okay, it seems like we have some more technical problems. I think while we wait for the speaker to resolve the problems, we'll go on to our next speaker. Well, the next person we have on stage is Mendi Letsoalo. And yeah, she's going to tell us how to grow Swiss chard and lettuce organically. Mendi, are you there?
Yes, I'm
Hello,
there.
Mendi. Okay, you can share your presentation.
Good day, everyone. My name is
Okay, I
Mendi
can see you. Okay, sorry. Yeah, you can continue.
Okay, can you hear me? Hello.
Hello, we can hear you. You can continue.
Okay, thank you. My name is Mendi Letsoalo from the University of Limpopo, and I will be taking you through a presentation on growing Swiss chard and lettuce on an organic medium in closed trough system, which improves growth yield and water use efficiency. Lettuce, scientifically known as Lactuca sativa, and scientifically known as Beta vulgaris, are common leafy vegetables consumed in South Africa for food security and economic values. These two crops are very sensitive to moisture stress, and they have a very high demand of nutrients. However, the accessibility of water for irrigation in our country remains a big challenge as a result of there is a need of a scientific knowledge on methods on how to adopt crop production methods which are efficient for water use. This figure one here shows an Omit system, and taking you through this system, it is a growing technique which is requires very low technological innovation, and it reduces the loss of nutrients and water through seepage. The Omit system is highly sustainable and less influence from the external environment, and it can accommodate a very high intensity farming. The aim of this study was to develop information on the growth and yield of lettuce cultivars and Swiss chard grown under the Omit system. The objectives of the study were to determine the effect of Omit system on growth and yield of lettuce cultivars Lara and Elisa, as well as to determine the effect of this system on growth and yield attributes of Swiss chard. Here is an Omit system and a non-Omit system on picture A and B. And this system, in preparing this system, this Omit system, soil was evacuated on a depth of 25 centimeters on raised benches, and the soil was pasteurized at 300 degrees Celsius, and the soil was mixed on a ratio of 2:1:1, 2 being loamy soil, 1 being sandy soil, and 1 being hygromix. A plastic was laid underneath the soil, and then the soil was added onto the plastic, and therefore another plastic was covered on top in order to make an enclosed trough. And then holes were done on 20 by 20 centimeters spacing. This experiment was conducted under greenhouse conditions at the University of Limpopo, and the experiment was arranged in a randomized complete plot design with the crops replicated 3 times. And seedlings were transplanted on the Omit system, and then the non-Omit system was used as a control. The parameters measured in this experiment was the biomass, the root length, the leaf length, as well as the stem diameter. The experiment on lettuce cultivars took place for 40 days, and then the experiment on Swiss chard was conducted for 63 days. The data collected on this experiment was subjected to a one sample t-test using a statistics 10.0 software at a probability level of 0.05. Here are the pictures of lettuce cultivars on Omit system and Omit system. As you can see from the pictures, Omit system encouraged enhanced a good plant vigor. Meanwhile, in the non-Omit system, the crops were not having a good plant vigor. This figure 5 shows the effect of Omit fresh mass and the total aerial mass. From the graph, we can tell that Omit system enhance a growth in fresh mass as well as the fresh aerial mass. And that has resulted in a very good biomass of the two crops of lettuce cultivars. Figure 6 here shows the effect of Omit system on root development of lettuce cultivars. On the Omit system, the roots were very long, and Omit the roots were relatively shorter. This means that the Omit system was able to enhance the growth of the roots. Figure 7 here shows the effect of Omit system on leaf length. Non-Omit system had relatively lower leaf length as compared to the crops system. This means that the Omit system was effective in making sure that the lettuce cultivars have an increased growth in the leaves. And we know that in lettuce, we normally consume the leaves. So this means that Omit was successful in enhancing the growth of our cultivar, our lettuce cultivars. Going on to Swiss chard, the pictures here show the effect of Omit. The effect of Omit on the growth of Swiss and also of non-Omit. As you can see from the pictures, the Swiss chard cultivar which was grown on Omit is relatively bigger as compared to the one grown on non-Omit. These two tables here shows the effect of Omit on the longest root length and on the biomass. In this case of Swiss chard, Omit resulted in relatively lower root length as compared to non-Omit. This is due to the fact that Omit was effective in saving water. Therefore, there was no need for the Swiss chard to grow deeper roots in order to access water. Therefore, Omit was effective in saving water for the Swiss chard. That resulted in a shorter root length. When it comes to biomass, Omit system resulted in a greater biomass of the Swiss chard, resulted in a relatively lower biomass of the Swiss chard. We see a table on the effect of Omit on leaf length and the effect of Omit on stem diameter. Omit resulted in a higher and a longest leaf length in Swiss chard, and that is very good because also in Swiss chard we consume the leaf. And when it comes to stem diameter also, the Omit system was effective in increasing the size of the stem, which resulted in, we can say, relatively longer and bigger leaves. Last but not least, and most important, the effect of Omit on cumulative water. Omit resulted a relatively lower usage of water as compared to the non-Omit. And Omit is very effective because it is not affected by the external factors such as evaporation. That means that more water was saved on Omit than on the non-Omit system. That is why it resulted in a relatively lower usage of water. The take-home message in this study is that the Omit system in both lettuce cultivars and Chard resulted in improved growth and yield. And farmers are recommended to adapt the Omit system growing technique in order to address the issue of water scarcity, because we know that in South Africa, we are having a challenge of water in the farms. The future works of this study, currently the studies are looking at nutritional compositions and secondary metabolites. I would like to acknowledge SAAB for a chance to showcase this work, as well as Department of Science and Innovation, NRF for funding, as well as the University of Limpopo. Thank you.
Okay, thank you very much, Mandy. Yes, we have a question here from the audience and from myself as well. Besides the plastic on Omit, what were the other major differences between the Omit and the non-Omit systems used? So basically what we're asking, what is the difference between the Omit and the non-Omit system?
As I've indicated, when we were setting up this Omit system, the only difference is plastic, because we're using the same kind of growth media and everything else is the same, so that the differences could only be due to the plastic that we use for Omit and not other things. If we use other different things, it means that the differences we see on our crops will not be as a result of the Omit system. It will be also the growth medias and other things which we might have differentiated in this setup.
Okay, so in other words, it's only the plastic that is differing between the two systems.
Yes.
Okay, thank you. All right, thank you very much, Mandy. We can continue to our next speaker. I see on stage we have Gaborone Tuni. Yes, yeah, he will be our next presenter. And yeah, he will talk about stress responses of soybean to salinity stress. All right, Gaborone, over to you. You can share your screen and unmute for us.
Hello.
Hello, Gaborone.
Can you hear me?
Yes, I can hear you.
Okay, great. I'm just sharing now.
Are you managing over there?
Yes, it's supposed to be on the way.
All right,
Please
there.
tell me when you
You see your presentation. So, Gaborone, you may continue.
Great. Let me just hide this. Good afternoon, ladies and gentlemen, esteemed guests, colleagues, and this is what I'll be presenting today: the morphological and physiological response of salinity stress on glycine max. I am from the Department of Botany at the School of Biological in the Noordwes-universiteit. This is an honors presentation in plant biotechnology, and let's see what we will be talking about today. I'll be sharing with you these outlined headings, and let us see what more do we have. Glycine max, also known as soybean, is used for nutrition, soil fixing, and biofuel production. Nutrition can be human nutrition, and it can be for animal feeds. The problem is that there are soluble salts in our soils that eventually affect crop morphology and physiology. Now let us see where are some of the sources that bring these soluble salts in the soil. Soluble salts can come from natural weathering, which is ultimately ubiquitous, and it is independent. Anthropogenic irrigation can also be another source. It is spatially variable and it's better managed. It is better manageable than the independent natural weathering. Soybean is mostly produced in 3 provinces, namely being the Free State, Mpumalanga, and KwaZulu-Natal. When soybean is being grown, salts have an effect, and whatever we do to ameliorate, conserve the crop can. Salt has an effect when the electrical conductivity of the soil paste is above 4 decisiemens per meter. There are studies whereby it is shown that 80 to 100 millimolar of sodium chloride can be equated to 4 decisiemens per meter. Now, at this level of salinity, there can be expected stunted yield and premature yield. Now, the thing is that with conservation of agriculture in general, it does not alleviate the stress. However, conservation can be achieved through measuring the soil salinity leaching and the electrical conductivity. We can also produce salt-tolerant varieties. We can also remediate the affected lands and also implement better environmental planning. One last thing that we can also do to conserve soils from salinity is by using organic or green synthetic fertilizers. Specifically, genetically improved cultivars can be our best solution, as they can be specially designed to be grown in different seasons. Ghana Seeds has presented us with a soybean cultivar recommendation for different regions that we have in South Africa. Specifically, we will be looking at the medium growth class under the cultivar name PAN 1521R. If you can take a look at the table, you'll be able to see that it is best adapted to all 4 regions, unlike the other cultivars. Now, this is the particular cultivar that we have used in our study. To take a look at further our literature review, soybean structure and function were taken into consideration because this is our model plant. So we therefore need to know better on how does it develop vegetatively and how does its morphology usually develop. It has a bush type growth habit with a pair of unifoliate and multiple pinnately trifoliate leaves. As we can see under figure 2, the growth stage, which is abbreviated VC, is the vegetative cotyledon stage, whereby it has the trifoliate leaves. No, it does not have yet the trifoliate leaves, but the first trifoliate leaf is currently developing, and we have fully developed unifoliate leaves. Now, soybean vegetative morphology is driven by genotype and stress. Specifically, the order of branching and the density or sparsity of the branching is driven by genotype and stress. The root expansion depends on the plantation culture, whether it is in a cultivated close range or a wild open culture. Growth and susceptibility is separated into either vegetative or reproductive stage. Now, this has an implication on the stress amelioration effects that can be expected when we conserve or when we add growth promoters to our crop, being that certain stress amelioration is most susceptible at vegetative stage. So it therefore means that we need to use similar terminology in order to aid better communication so that we have better results at the end. Now, counting of the nth node on the main stem is how we determine at which stage is the crop. After the vegetative cotyledon stage, it means that there comes the V1 stage, meaning that it is the first vegetative stage after the vegetative cotyledon stage. Also the VE, to elaborate what the acronym means, it's the vegetative emergence stage. So going forward, we will be counting the ends up until where is necessary. Continue, we will be taking a look at the nutrient transport pathways. Nutrients and water uptake uses xylem driven by transpiration and root pressure. Phloem translocation. Phloem translocates amino acids, photoassimilates, and absorbed minerals. This can be seen on figure 3, whereby it shows unidirectional flow of xylem and the bidirectional flow of phloem. Salinity alters nitrogen-specific amino acids, which help in transportation under the xylem disc. Salinity also alters the osmotic pressure or protoplasmic streaming used for phloem transport. Let us take a look at the morphological responses of soybean to salinity stress. Here are the salt-affected agronomic traits which soybean is considered to be affected, which under soybean are considered to be affected. Biomass accumulation, plant height, leaf size, number of nodules, internodes and branches, weight per plant and weight per seed, protein and oil content, and ultimately the yield productivity is affected. Now, these agronomic traits are what is necessary for a crop to be of value. The morphological, physiological, molecular, biochemical, and genetic information used with genotypic responses can be ultimately understood in order to confer salinity tolerance. Soybean under salt stress is dependent on species-specific growth stage. So hence it is important to be aware of the specific communication aids or the specific growth stage aids that we would be using to communicate at which stage is soybean. Also, the cultivar or the genotype and environmental stress eventually affects how soybean eventually affects how soybean responds under salt stress. That soybean is susceptible to 4 decicem to ECE that is greater than 4 decisiemens per meter. However, another study found a threshold of ECE that is above 5 decisiemens per meter. This shows that salinity stress is differentially understood, it is differentially responded to by different cultivars. So at the end of the day, we need to stress and test as much cultivars as possible. How does salinity affect growth and development? Plants grown in different habitats and life cycles respond differently. This is a three phases. This is a 3 phases image that describes plant growth, development, and survival under simulated salt stress effects. From stress onset, what we can clearly see is a sharp decline in productivity, whereby the phase 1 is characterized with a rapid water deficiency. And the second phase is characterized by ion buildup. Ion buildup now is where tolerant and sensitive plants start differentiating, whereby tolerant plants will continue at their current trajectory, and the sensitive plants, being glycophytes, as a glycophyte, it would be deteriorating in productivity. Now, the third phase is intertwined between phase 1 and 2, whereby it is represented by reactive oxides production. Let us take a closer look at these 3 stages. Salt is diluted by dividing cells. Salt is differentially responded to by new, meaning young leaves, and aged leaves. New leaves are much more tolerant than mature leaves. Tolerance to cell damage varies among chlorophyll content and photosynthetic systems are altered. These 3 phases affect these growth and development responses. Let us look at the physiological responses of soybean, whereby specifically the phase 1, 2, and 3 we will.
Hi, Gabaran. You have about 2 minutes to wrap up your talk, please.
Okay, let us take a look at efficiency, whereby of electrochemical gradients of the cell receptors allow us to see which antagonistic ions are at play. The symplastic and apoplastic flow have selective channels. Vascular pathway receptors in the plasma membrane optimally perform work, suboptimally perform work. Osmotic stress is the loss of water potential, meaning that there will be a decline in leaf water content. Hypotonicity between the cytosol and vacuole reduces cytosolic water activity, and the is actually not being able to pass osmotically due to the osmotic gradient. Now, ion stress is due to the sodium chloride toxicity, whereby it depends on the proteins and electrolyte concentration in the cytosol, stability of the protein complex in receptors, charge density, and individual ion properties. It also affects soybean because it is toxicity affects leaves susceptible soybean that already excludes sodium. Ions dissipate energy for work, meaning that adjusting the charge is needing regulation. Ion compartmentalization is at play instead of a direct on of the ion on a protein. Oxidative stress is part of cellular metabolism and can be found in the cell wall, plasma membrane, mitochondria, chloroplast, and perosomes. ROS stress affects lipid peroxidation. It damages the DNA, proteins, and carbohydrate metabolism. It also leads to apoptotic necrosis. And antioxidants and non-enzymatic oxidants are also included. Salinity caused climate change. Salinity driven by climate change and degrading resource use is at play. In semi-arid regions, glycine max is produced in low quality, and in South Africa, salinity affects 100,000 hectares. Salinity affects important protein synthesis. Salinity also shrunk 23% of soybean production. The current operation of crop yield or crop production is degrading in South Africa. Land conservation technologies are under low adoption by smallholder farmers. And in our study, our aim is to evaluate the physiology and morphology of soybean. Our objectives
are.
Yes,
I think you've gone way over your time with your presentation. Can I maybe just check from our technical support a time check? Can we give Gabron another minute or two, or shall we go on to our next presenter?
I think let's give him 1 minute to conclude his remarks and
Okay,
then
Gabron, can you just go to your conclusions and conclude with your results, please?
Okay. Yeah, our results, plant height was statistically significantly increased in the moderate treatment. Shoot and root lengths were significantly increased. Also, our shoot and root weight were much into it. Water content was significantly increased. The chlorophyll content reduced. The stomatal conductance was significantly inhibited, and ultimately also our photosynthesis was significantly inhibited. Stress is interactive in different responsive pathways. Our morphological traits are heritable, so they are best for assessing environmental stress and also phenotyping plant vega. Leaf discoloration was due to generally the nitrogen that is antagonistic, that is antagonized by chloride, and the potassium was antagonized. Leaf area that was increased was due to an adaptation to low tuber pressure. But however, it was differentially upregulated at 50 millimolar, which is moderate than at severe treatment. The stomatal number was significantly increased in severe treatment. The plant height was increased due to osmotic regulation, ion dilution in growing tissue, and ultimately plant tissue tolerance. The shoot length was increased significantly in severe treatment. And this can be from a positive water potential gradient across the cell membrane that is established to restore turbo pressure. Relative water content surprisingly increased an osmotic adjustment, and our stomatal conduction was significantly inhibited. This was due to affected gut cells that were caused so by an effect by antagonistic sodium. This rate was strongly inhibited in severe treatment. Stomatal closure and stomatal our physiological response varied as salt concentration and the traits were affected by salinity, and the soybean cultivar some tolerance mechanism, physiological, morphological trait, and parameters basically actually most were tolerant. However, oxidative stress was most destructive. The future work of our study is to provide extensive assessment of comparative genotypes under differing environment stress. Our results provide a vital background for this cultivar, and also the results can be used as a recommendation soybean genotype screening on Salinity. All the partners and parties that played a role in my research, and to all of you for giving me a listening ear. So I went a bit over time. It's more than appreciated.
All right, thank you, Gaborone. I don't see any questions in the chat box. Maybe just one quick fast question from my side. Is salinity stress a major problem for soybean production in South Africa?
Yes, it would. There is only one cultivar that is specifically tolerant to a variety of environmental stresses. Now, salinity being that it is rapidly increasing, it means that it is a problem. Even though this is unverified, because there have been studies being done in South Africa, I believe so. The literatures that I have read, and for the mere fact that 100,000 hectares are affected by salinity in South Africa.
Okay, no, thank you very much, Gaborone. I'm looking forward to see more research from your side. All right, on to our next speaker. I believe our next speaker is Annelieswe Makatini. I saw her now. I think she is on stage. And she will be giving a talk about fungi decontamination protocols. All right, over to you, Annelieswe. You can share your presentation with us. Annelieswe, are you there? You can share.
Yes, I'm trying to share screen too.
All right. Okay, thank you. We can see your presentation.
Okay.
Yeah, if you're ready, you can continue and start.
Okay, thank you, sir. Ladies and gentlemen, good day. I'm going to be presenting to you a topic titled Development of Fungi Decontamination Protocols for Trichinella trigeniana seeds, a representative of recalcitrant seeds. I'm trying to move to the next slide and it's not happening.
Elisa, if you use your mouse and you click on the bottom left-hand side of your presentation, you'll see little arrows appear. You can advance your slides there. Bottom left-hand side?
Bottom left-hand side.
Bottom left, by the beans. Just hover your
By
mouse.
the beans, okay.
Okay, click there.
Double click.
No, just click once.
I am, but it's not moving.
Okay, does it show a little arrow at the bottom if you hover over it?
No, ma'am.
Would you like us to share your slides on your behalf?
Okay.
Just click on your presentation page once and then move with your arrow down and see if it will advance.
Once and no.
Okay, and if you press enter, it's not moving. All right, just press stop share and then we will share on your behalf.
Okay.
Just give us a second to bring the presentation up.
Okay, thank you.
Great.
Can you please move to the next slide? Okay, this is my content layout. Moving on to introduction. Difficulty to collect and store recalcitrant germplasm of tropical and subtropical plant species is a major problem. Why is this? Recalcitrant seeds are shared at very high water contents, can be stored intact in hydrated state for short to medium term. Deterioration of these seeds in hydrated storage is frequently encountered. Hydrated storage conditions also are conducive to endogenous contaminants. Thus, the proposed suited solution is understanding the type of microbes that live in and on this type of seeds and ascertaining antimicrobial agents effective against these microbes with minimal impact to seed physiology. Next slide. The aim was to isolate and identify fungi in recalcitrant seed that proliferate at culture stage to assess the efficacy of biological and chemical treatment on identified fungal isolates. And it was hypothesized that assessment of the antifungal chemical and biochemical activities may assist in the development of protocols for medium or long-term storage of some recalcitrant seeded plant germplasm. Next slide. The objective: to screen of freshly harvested recalcitrant seed of Trichinella georgiana for internal contaminants, that is to detect, localize, isolate, and identify common seeded associated fungi. To assess efficacy of biological and chemical treatments in curtailing or eliminating proliferation of selected fungal isolates using the agar plate technique. To assess and develop treatment protocols from 2 above for seeds prior to hydrated storage. To assess the efficacy of developed protocols in terms of survival of inherent microflora, effect on storage longevity of the seeds in hydrated storage, and lastly, to assess effects of fungal control treatments on seed germination and seedling vigor in terms of percentages of seedlings produced showing both root and shoot development, plant height, leaf area, and dry mass. That is looking at the dry mass or biomass of root, stem, leaves, and cotyledons. Materials and methods. The isolation and identification of fungal contaminants. Fruits were collected from Durban and surrounding areas in KwaZulu-Natal. They were then transported to the lab. Seeds were excised from the fruits by removing the waxy areole, thus exposing the green cotyledon surfaces. These were collected into a beaker and kept moist by enclosing with a thick layer of moist paper towel. Once enough numbers were attained, the seeds were then surface decontaminated using 1% sodium hypochlorite for 10 minutes, after which were rinsed 3 times using sterile distilled water. They were then allowed to dry overnight within a thin paper towel on a bench top under ambient temperatures. They were then stored under hydrated condition in a decontaminated 5-liter bucket at constant 16 degrees Celsius. They were then sampled at days 0, 1, 2, 4, 6, 16, 34, and 64 days interval in order to assess viability of seeds. However, this served a double purpose, as any fungal contaminants that prevailed were recorded, isolated to obtain pure axenic cultures for identification. Next slide. As mentioned in the previous slide, any fungal proliferation that emerged in hydrogen storage or in tissue culture were isolated and immersed in quarter-strength Ringer's solution and agitated for 10 minutes at 150 RPMs. The 10 ml aliquot of this solution was serially diluted up to 10-6, from which 1 ml was mixed with 30 ml of potato dextrose agar before setting. The cultures were incubated at 25 to 30 degrees Celsius for 5 to 10 and monitored daily. Please, this I note, 25 to 30 is a range because these were incubated at ambient temperatures. So the temperatures varied from 25 to 30. As mycelia developed, individual plugs were subcultured into fresh potato dextrose agar and incubated. This was repeated 3 times as to ensure axenic cultures. The cultures were examined with a stereomicroscope, and the frequency with which a genus occurred was expressed as the number of isolates of the same genus divided by the total number of genera isolated per that plant species. In this case is Trachea trigena. Moving on to characterization, plugs of axenic cultures were introduced into 9 ml of Ringer's solution and vortex to disperse the spores. A sterile needle was used to collect an inoculum, then plated on each 5 different media, namely malt agar, suborate agar, potato dextrose agar, water agar, and Zapex agar. Those are represented as picture number 2 on your presented slide. Fungal ID was carried out microscopically by inspection of colony margins, surface and underside textures, pigmentation, and growth over 7 days. The prepared slide were viewed and characterized at low and high magnification. Images of fruiting bodies, structures were captured and identified according to wrapper and funnel. Molecular identification was accessed with Ngaba Biotech to confirm these findings. Please move on to the next slide. Right. The slide is basically showing all the isolated fungal isolates from the Trachea trigena. Please note that 6 fungal species were identified as per outline on the slide, and then P. lilacinus was the most prevalent fungus occurring at a frequency of 0.55 on PDA, followed by the other identified fungal species, and you can see each of those, the frequency of occurrence was at 0.09. Please move on to the next slide. Then this slide basically shows us the assessment of biocontrol agents that were chosen from a long list of biological controls that were first or initially started with, and these were EcoT, Eco77, and B. subtilis. Please note that B. subtilis was the least effective antimicrobial agent when compared to trichoderma strains of EcoT and Eco77. However, when comparing the 2 trichoderma strains, no differences were noted, but had a slight difference when the actual inhibition zones were compared with when the actual inhibition zones were compared with Eco77, exhibiting slightly bigger zones of inhibition. In addition, the most problematic fungal genera in storage is that of Penicillium and Fusarium, and it is noted that with EcoT, we achieve an inhibition zone of 45 with penicillium, and then an inhibition zone of 26 with fusarium. Although ECO77 was more effective than the other tested biological agents, with both biological agents, both strains of trichoderma were also considered for further development of seed treatment protocols. Please move on to the next slide. Then the assessment of chemical control agents. Please note that from this list of chemical control agents, the results indicated that nipastat, Orias, and biotin were the most effective chemical control agent, with repetinate being the second last at the end, repetinate being the least effective, based on the data presented in tables 3 and 4, seed treatment protocol based on ECO-T, ECO77, nipastat, biotin were established. Please move on to the next slide. Okay, this slide basically is projecting the developed protocols. These are the developed protocols where DBN, represented as the first on the list, is serving as a control. Whenever you see a tick, basically it means the protocol incorporated the antimicrobial agent to treat the seeds before hydrated storage, and then a minus would mean an exclusion. As projected from the slide, basically, on the top column is in the first row, we have ECO77 biocontrol at 1 gram per liter. You can see as you go down as the first treatment, DEBBE, you have a tick there. Basically, it means ECO77 was incorporated while developing that protocol, same as biotin, same as benomyl, excluding nipastat and encapsulation. So basically, if you read through that, I will have like a key indication in each of the tables that are going to be shown. Please move to the next slide. Now, results and discussion. Table 5, this is table 6, shows the results from material that had been hydrated stored at 67 days and then assessed for 30 days in tissue culture. Treatment DEBBE, DEBN, and then showed no contamination. Can you please move on to the next slide? Showed no contamination. Treatment then had a significantly high root and shoot development, which was at 100% of explants produced shoots and roots, whereas DENN was considered the best treatment to maintain or to improve seed lifespan in hydrated storage. Please move to the next slide. Okay, leaf area. Basically, I'll make cross-reference to the first table, which was table 5 in this case. So table 5 and 7, both seed treatment and storage time had a significant impact on leaf area, with mean leaf area values ranging from 291 to 609 square millimeter. In particular, the longer storage period, the greater the impact. In this regard, the leaf area of the control, which is at day 0 of treatment DBE, was 609 square millimeters, but for the material that had been stored for 64 days, it had decreased to 166 square millimeters. Moving on to the next slide, which is leaf biomass. Leaf biomass tables 5 and 8, seed treatment and storage had no significant impact on the leaf biomass. The average leaf biomass of the control was 0.49 gram at day 0, whereas the other treatment ranged from 0.6 to 0.68 gram. Control treatment DBE at storage day 67 had the lowest leaf biomass of 0.15 gram, whereas treatment DEBN had the largest leaf biomass of 0.34 gram. When compared with the control treatment, DBE, and the other treatments. Therefore, it was concluded that the leaf biomass was neither negatively nor positively influenced by these factors. Please move on to the next slide. Root biomass. Both treatment and storage, regardless of each of other, had a significant impact on the root biomass. However, where the two factors interacted, no effects on the root biomass were observed. Seed treatment with DBE at day 0 had root biomass that averaged at 0.61 gram, with the biggest biomass being obtained with seeds that were in storage for 2 days and 4 days with treatment DEBBE and DEBN, which both averaged at 0.7 gram. Seeds that had been initially exposed to 64-day storage produced plantlets that had the smallest root biomass of 0.25 gram for controlled treatment DBE, whereas the other treatment had a range from 0.36 gram to 0.55 gram. Treatment DEBN had a positive impact on root biomass when considering time of storage at 64 days, even though the seed appearance after treatment was not good. That is, basically the seed had brown patches. Impact observed for seeds treatment with treatment DEBBE was the same for days 1, 2, 4, 6, 16, and 32 days. Please move on to the next slide, which is stem biomass. Stem biomass was significantly affected by the seed treatment and storage. At the start of the treatment, the control stem biomass averaged 0.50 gram, whereas the other treatment, DEBBE, DEBN, DEN, and DENN, ranged from 0.22 to 0.4 grams. It appeared that increasing storage period had a positive impact on stem biomass, with the largest stem biomass of 0.64 gram being obtained from DEBBE, treated seeds stored for 32 days, followed by treatment DEN, treated seeds stored for 64 days, which had a total dry biomass of 0.51 gram. Please move on to the next slide. Conclusion. The study has not only shown the detrimental impact of seed-associated microflora contaminants on seed viability, but also significantly demonstrated the following: the efficacy of nipastat, biotin, and Eco77 as a decontaminant for these seeds, and potentially other types of explant of subsequently short to medium term storage and other purposes. Exposure to nipastat, biotin, and Eco77 has a potential for a complete elimination of seed-associated field and storage fungi with no diverse effect on seeds or explants. Alternate encapsulation with or without nipastat showed significant potential in eradication of field, storage fungi, or opportunistic contaminants via cross-contamination and for the formation of syn seeds. Lastly, the study has marked a practical impact for both short and medium-term storage of Trichinella Drogeana seeds. Please move on to the next slide. Recommendation for future studies. It is recommended that they develop protocols for the elimination of fungal agents associated with recalcitrant seed of Trichinella Drogeana, including either the encapsulation of seed in calcium arginate or seed treated, a combination of chemical nipastat and biotin, and biological Eco77 agents without any apparent effect on the viability and vigor of seed or saplings to be further assessed at microscopic level. And then hydrogen storage was assessed up to 64 days only. It would be interesting to extend the period in order to ascertain the impact, if any. Please move on to the next slide. These are my acknowledgements. I acknowledge NRF for funding the studies, my colleagues, and as well as ESS, my presentation. Next slide. These are my references. Next slide. Thank you.
Okay, thank you very much, Annelieswe. Are there any questions for her? If you want to, you can quickly write there in the Q&A. I can just ask, what is the economic importance of your research?
For economic importance, basically for the R&D based companies, and as well as for the plant germplasm storage or tissue culture based facilities, it would really assist them as in to save money when these materials are collected from the field or different field. And you know, this material is recalcitrant seed and they are very problematic to actually store. So to be able to store them for longer, for usage, for research purposes, or for plant germplasm conservation, they will benefit a lot in order to eliminate all the contaminants that might be occupying the seeds, other than to store and then to find that you have stored in tissue culture and you lose everything, as in the whole bunch of tissue culture work that you have preserved would basically go to waste. But then if the fungi is curtailed at initial stages, at least you are certain and you know for sure that your material will be able to be stored and you can continue with your research.
Okay, yeah, thank you very much, Annelieswa. All right. Let's see, who do we have on stage? Is Gumbo, has he managed to get on? Okay.
Gumbo, unfortunately not. We can maybe try, I'll let you know if he appears, maybe Daniela.
Daniela, yes, okay.
Danielle, if you're there, if you can just activate your camera and microphone for us.
Yeah, okay, yeah, there's Daniela. We lost her earlier because of a big storm and
Yeah,
a power
sorry, my
outage.
power went out for half an hour. It came on just now, luckily.
Luckily.
Yeah.
All right, okay. Danielle, you're welcome to start again. So over to you.
Okay, can you see my screen in full presentation mode?
Yes.
Hello?
Yes, we can see it. You can proceed.
Okay, thank you. Okay, my name is Danielle Botha, and the topic of my presentation is an RBCL and tRNL reference database for the identification of food items from herbivorous diets based in the South African savanna. This research project was completed with the help of my supervisors, Professor Chandra Barnard, Professor Francis Siebert, and Dr. Mornay du Plessis. Sorry. The research I am presenting is an important aim I have fulfilled for my dissertation, which is about bioinformatic filtering options in herbivore dietary analysis following a DNA metabarcoding approach. In order to identify traces of DNA left in the feces of herbivores, one firstly needs an accurate DNA reference database to compare the unknown sequences against. This DNA database is compiled by acquiring species lists representative of South African savannas, which will then be used to download their corresponding sequences. This database must also undergo some testing and statistical analysis to be sure that this is effective in species identification. The importance of a custom DNA reference database, as well as the fulfillment of the objectives, will become clear throughout this presentation. I wanted to start with this figure from Siebert and Dreber to illustrate the amazing diversity we can find in South African savannas. Imagine being able to take a sample of soil or some herbivorous feces and characterizing specific components of the plant community. This is the idea behind environmental DNA, and that all living organisms leave traces behind in their environment. But how do we identify an organism from environmental samples? This is where DNA reference databases come in, and why they are so important. eDNA from the environmental sample is PCR amplified with a certain barcode and sequenced with next-generation sequencers. These barcodes are then compared to a global library of taxonomically informative barcodes to find out what their host organisms are. GenBank and BOLD are examples of such global DNA libraries or DNA reference databases. It is well known that global sequence databases are not perfect and are often incomplete, unverified, irrelevant, and contain sequences of poor quality. When using them as is, you may introduce erroneous and unverified data into your study. By creating a curated subset of this data and supplying your own sequences, you boost the reliability of your study considerably by eliminating inappropriate comparisons. The accuracy of a meta-barcoding study is therefore underpinned by a complete and accurate DNA reference database. The species used in this study are chosen to acquire the most accurate representation of South African savannas. The species could be sourced from other studies conducted in areas and game reserves, such as the Kruger National Park, as well as a study on savanna land use type, focusing on Valverdiend on a communal rangeland, as well as Syferkelp Farm in Limpopo. The study by Gill et al., which was more applicable to African savannas, published their complete database on BOLD and GenBank, which was filtered by an expert botanist to recommend South African species addition to this study's final species list. The species list is compiled on the BOLD workbench as a checklist and will look like this after you have supplied all the necessary taxonomic information. It is very convenient as it gives you a breakdown of the taxonomy as well as supplying a DOI which you can include in a paper. From here it can be seen that my species list consists of 731 species from 99 families in the phylum Tracheophyta. A disagreement report is also available to ensure that the provided taxonomy is correct. Here it can be seen that there are no taxa with hierarchy disagreements. A progress report of this species list also indicates how many sequences have barcodes on BOLD. This is very useful when downloading sequences and checking their availability. Sequences for species from the species list, after they have been downloaded from global databases, they are downloaded according to certain criteria. The species must have a voucher specimen. Individuals for each species must ideally not be from the same herbarium, and a maximum number of 3 individuals are downloaded for each species per marker. The resulting downloads consist of 1,159 RBCL sequences and 850 RNL sequences. 109 of the species on the species list could not be found on any global database as of November 2020. Okay, now that you have downloaded your sequences, they are aligned with muscle using Genius Prime software. The RBCL sequences were aligned simultaneously, while the tRNL sequences were aligned by family due to this barcode exhibiting highly divergent sequences among distantly related species. Neighbor joining trees are built using Camera 2 parameter genetic distances, and the accuracy of the phylogenetic tree may be boosted by visually inspecting the trees to see if reasonable groups are formed, such as shrubs, grasses, trees, and forbs. If a significant outlier was detected, it was replaced with another sequence fulfilling the same criteria, or it had to be removed completely. Figure A is an example of the neighbor joining tree retrieved from the RBCL sequences after they were downloaded. The tree was then checked by an expert in the field of botany to eliminate species which did not group well and could be replaced by a different sequence or removed. Figure B represents the updated phylogenetic tree. It would be impossible to include the whole trees in this slide, but from just the top of the trees, one can already see a big positive difference. After the informed removal of 60 species, the replacement of 59 individuals, and the addition of 60 species from the Gill et al. Database, species grouped much better, and the bootstrap values of the overall trees indicated a much greater confidence in branch placement. The same trend can be seen in the neighbor joining trees of the tRNL sequences. The data analysis of the DNA reference database entails determining the presence of barcode gaps, determining a data-appropriate identification threshold for the tRNL and RBCL datasets, as well as using various distance-based matrices to assess the identification efficiency. Let us start with barcode gap analysis. It is the assumption that the amount of genetic variation within species is smaller than the amount of variation between species. This allows the two to be distinguished. As stated by Meyer et al., the barcode gap should be calculated using the smallest rather than mean interspecific distances. The barcode gap analysis was performed for RBCL and tRNL datasets separately. The first step is the calculation of genetic distances via the function disDNA in the APE package. The output of this is a pairwise distance matrix calculated with Chimera two-parameter model of evolution. The second step is to generate two statistics for each individual in the dataset, the furthest intraspecific distance among species, as well as the closest non-conspecific or interspecific distance. The output of this subtraction may then be stored in a data frame where barcode gaps are indicated with a 1 and the absence thereof with a 0. The barcode gap analysis was only possible for species represented by more than one individual. The scatter plots on the left side of the screen represents sequences before the phylogenetic trees were checked and sequences were removed, and the right side indicates the new database. 71% of RBCL species and 75% of tRNL species exhibited a barcode gap in the old database. 77% of RBCL species and 71% of tRNL species exhibited a barcode gap in the new database. More species are also represented in the new database, since many species could be added from Gill et al., which are also representatives of the South African savanna. One can clearly see that the new database would show greater success in species discrimination, since more species exhibit a barcode gap, as seen by the density of species grouping above the 1-to-1 slope. The bold identification threshold is 1%, and this is not always best fitted for species identification of your dataset. Importantly, for data where unsampled species are likely to be encountered, like in this dataset, it is important to optimize the threshold to ensure accurate identifications. The optimum threshold is determined by the threshold function in the spider package. And by looking at the sense mat on this graph in front of you, we can see the optimum threshold as reflected in these graphs. The optimum threshold is the lowest cumulative error, which is the lowest amount of false positive and false negative identifications. False positives are identified by the light gray bars, and the dark gray bars indicate identification error rates summed across a range of distance thresholds from 0 to 2% in 0.1% increments. The optimum threshold for the RBCL old database is 0.2%. For RBCL new, it is 0.1%. For tRNL old, it is 0.5%. And for tRNL new, it is 0.4%. The identification accuracy of the reference database is determined using three primary distance-based criteria: Meyer's best close match, nearest neighbor, and bold identification criteria. These methods simulate an actual identification scenario by treating each sequence as an unknown query and the rest of the database as your reference database. The use of these identification scenarios may not always be an accurate reflection of the efficiency of the reference database, but one can get an approximation of identification success rates. The new database showed much higher success rates for species discrimination than the old database, which is circled with orange. Among the distance-based criteria, the nearest neighbor criteria fared the best, since these methods ignore uncertainties in cases of a tight closest match and will offer identification based on the majority rather than classifying sequences as ambiguous, as in Meyer's best close match and bold identification criteria. Conclusions I could make from this study are that it was possible to utilize species lists from ecological surveys to compile RBCL and tRNL reference databases to identify food items from a herbivore's diet foraging in an South African savanna. Global reference databases could be extensively mined to gather individuals for each species per barcoding marker to build a potentially useful resource for plant identification from environmental samples in a South African savanna. These reference databases could be optimized and evaluated for their potential efficiency for species delineation. And lastly, the importance of taxonomical expertise is highlighted input from botanical experts, showing overall higher success rates of species identification. Here are my references. Thank you so much for listening.
Okay, thank you very much, Danielle. Are there any questions from the audience? I'm sure there must be one. No questions. Yeah, I don't have a question either from you. These samples, they were collected in the Kruger National Park, if I'm not mistaken, or where did you?
Yes, it's not the samples that were collected, it was the species list that we used to download them. The raw data that I will eventually compare against this reference database is from a communal rangeland in Belvedere and then the Syferkuil farm in Limpopo.
Maybe a stupid question, but why did you collect it in animal feces and not use felt samples?
We wanted to see in especially the cattle diets. I don't know if everyone knows this, but FORBs are very weakly presented on databases and in management models. So we wanted to see across different land use types what the cattle prefer to eat, what was more dominant in their diets, and also to see the significance of the FORB species to then add them to databases and literature.
Okay, no. Thank you very much, Danielle. I don't see any other questions. All right, yeah, thank you very much. You can unshare your screen, please. Let's see, I don't think Gumbo is on stage. I doubt it. Angela, can you maybe just confirm for me? Chat. Not yet. All right. Okay. Then this session has come to an end. I want to thank all the speakers, and yeah, we can take a nice comfort break now, and we will continue at 20 minutes past 3. Okay. Goodbye.